everyone for attending today and for the invitation to present. And the title of my talk is Sex-Related Differences in Colorectal Cancer Metabolism and Patient Prognosis. So I'm going to be presenting a little bit of our results from our untargeted metabolomics and actually how we use that information to investigate the biology of our phenotype. So there's a lot of in vitro and in vivo work that's sort of followed on from that right now. And the same with Dr. Graham's study. This is about a 5 or 6 year project really that we've been building on and we're still building on right now. So I just wanted to mention what the focus area is of my lab. So we're really interested in understanding both the genetic and environmental influences on colorectal cancer progression. And that includes aspects such as the microbiome, diet, environmental pollutants. And what we really want to find out is any metabolic profiles that could be associated with various cancer phenotypes. So in order to do this work, we have our workhorse mass spec, which is our Waters Zevo QTOF, and I've had this instrument for about 6 years now. And on this instrument, we've developed numerous workflows. We have a general comprehensive workflow where we look at, we use both a helic and reverse phase analysis to look at those polar metabolites and non-polar metabolites. So we try to cover a wide breadth of metabolism as you possibly can. And when we find our metabolites of interest, we then look to see how they're related to each other in metabolic pathways and in signaling pathways to try and get to the biology, which is sort of underlying these phenotypes. And then we like to also relate these obviously to the clinical relevance. So our main area of interest within the lab is the colorectal or colorectal cancer. And as you see, I've been quite handy on biorender here. So this is where I obtained most of these pictures. But you can just see by looking at this organ, it's very large and it's actually very heterogeneous. So there are various regions of the colon that start from the cecum, which is on the right side near the appendix, going up to the ascending. And then you have the left side of the colon, which is on the descending side, and also the sigmoid colon and the rectum. And what we've seen in many different meta-analyses now is that if the patient has a tumor which occurs on the right side of the colon, they have a much poorer prognosis, and if it occurs on the left side of the colon. And many studies have, looked at this issue, and they've, adjusted for many confounders such as screening, when what stage the tumor was identified, the type of therapy given, and they still see that patients with right-sided colon cancer have a poorer prognosis. What's interesting about this as well is if a patient has a right-sided tumor, they're more likely to be female, which is quite different from what you see for most cancers, where males tend to have a higher incidence if the tumor is in a non-reproductive organ. So to me, this was really quite striking why there was this difference. And then I wondered too whether there could be some strong metabolic differences that underlie these different regions of the colon. In addition to this, there's also obviously differences in mutational frequencies. So on the right side, you have more KRAS mutations, BRAF mutations. On the left side, more APC and TP53. There was also a nice paper out a few years ago that came from Harvard where they looked at different lifestyle exposures and where the cancers occur. And they found that smoking and alcohol use was associated more with the transverse colon cancers, whereas decreased fiber intake and Western dietary patterns and a larger waist to hip ratio was associated with sigmoid and rectal cancers. And then one of the interests in my lab had been to look more at the differences in microbiome and also metabolites that occur in these areas of the colon, because they can also be linked to cancers in these areas. So one of the first studies I did actually during my postdoctoral work in collaboration with Johns Hopkins University was to look at the bacteria in these different areas of the colon. And my collaborators found bacterial biofilms, which were located on the right side of the colon only. And this is basically sort of dense aggregates of bacteria that are encased in a polysaccharide matrix. And we found that they were causing increased proliferation and inflammation in that side of the colon. And there was also an associated metabolic phenotype, which indicated proliferation as well. So lots of polyamines increase and things like that. More recently as well, I'm not going to discuss this paper, but there's a reference here from Tsai et al., where we also saw differences in microbial metabolites, the bile acids, in these different areas of the colon. So they're much higher on the right side. And we also see differences in conjugation of these bile acids by sex of the patient as well, and they associate with inflammation and prognosis. So you can just, I think I just wanted to outline just how heterogeneous this organ is. And a lot of metabolic studies have been done using blood from these patients, or they haven't really looked at subsites of the colon. And I think this is really important to stratify down to really understand, the differences in biology in this very large organ. And I just wanted to touch again on the sex differences. So this is a review paper from a few years ago now, which just shows the differences in incidence and mortality for various cancers from different locations in the body that are from non-reproductive organs. And the darker blue, if you can't see back there, is female incidence, and the lighter blue is male. You could just see that males generally do have a higher incidence of cancers compared to females. So it's quite interesting why there is this preponderance in the right side of the colon of a higher incidence in females. There have also been some differences seen by sex in terms of therapeutic responses in colon cancer. So this study from a couple of years ago, they looked at differences in responses to either a 5-fluorouracil and bevacizumab treatment, or a combination treatment which also included irinotecan. And on the left side of this diagram, you can see there is a better survival for male patients that received the combination treatment, whereas females actually did a lot worse, and they were better off with the original 5-fluorouracil and bevacizumab treatment. So you can see there are differences just by sex of the patient, again, in how they respond to treatments. But typically, sex isn't really looked at when prescribing a drug for cancer, to my knowledge, in colorectal cancer. So, but I think this is very important to look into as well. So getting back to these sex differences within colorectal cancer, what could really potentially underlie this? Well, of course, we have very different lifestyle exposures in our body. So there are effects from hormones, of course. There's differences in lifestyle, even things like pregnancy is known to change your metabolic profile. There's differences in medication between males and females, oral contraceptives, HRT, of course, potentially differences in microbiome and antibiotic uses, and differences in environmental exposures, through different occupations and things like that. And, as Dr. Graham alluded to as well, all these different factors can really change your metabolic profile. So it's possible there is differences in the metabolic profile between males and females with colorectal cancer. So we had 3 major questions that we wanted to answer in our research. First was, does a sex-specific metabolic phenotype exist for colorectal cancer? Are these metabolites potentially prognostic? And what are the genetic and non-genetic influences that could really affect or underlie these associations? So you may recognize Nick Stewart, who works as a collaborator. So the first study we did was using a biobank of tumor tissues that we obtained from Sloan Kettering Cancer Center in New York. These samples were really a goldmine for us to obtain because they were excised from patients who were given surgery for colectomy in the 1990s. And what they did was they placed the tissues in liquid nitrogen, and then they put them in the -80. So they were in the perfect condition for us to do metabolomics. What we found, when I initially got to Yale, was that a lot of the tissues have been, formalin fixed, put in paraffin blocks, which we can't use for untargeted metabolomic analysis. Another thing with these samples is that they were collected in the 90s. So we also then had the follow-up information. So we obtained a grant to go to the medical vault at Sloan Kettering and pull out all this information about the patients, and even things like their occupation and things like that, and their follow-up, their prognosis. So in order to just stratify a little bit more closely to begin with, we just looked at the left and the right sides of the colon because we knew there were these differences in incidence, and we didn't know what we would find initially. So we had about 200 tumor tissues and associated normal tissues as well. And we performed untargeted metabolomics. And Dr. Rattray and Dr. Sai really did all of this work. So I just wanted to acknowledge them here. And they've now gone on to better things. Actually, Nick is now a tenured associate professor at University of Strathclyde. I have a close collaborator on this work as well, Dr. Khan, who is in Department of Surgery at the School of Medicine, and he really guides us a lot for the clinical translation of this work. So this is just really in brief our workflow. So we had these normal tissues and tumor tissues. We extracted them for metabolites, and then they underwent comprehensive metabolic profiling. So, and we had at the time just a small metabolic database of about 100 compounds, but we've now built this up to about 1,000. So we're able to do some instant identification of compounds. And then we looked at the sex differences in these pathways and looked at the associated survival of these metabolites, related to these metabolites, sorry. So you probably can't see this very well all the way back there, but what we found initially was quite striking to us. There were lots of differences in many different metabolites between males and females. So what we have here are numerous box plots where in the first box plot in green is the normal tissue, and then we have stage 1, 2, and 3 tissues. And these are different metabolites within these tissues. And on the right side of this diagram is 3 metabolic pathways. We have glycolysis, pentose phosphate pathway, and fatty acid beta oxidation. And what we saw was in general that in the tumors from male patients, the glucose is primarily oxidized into lactate through glycolysis. And what we saw in the tumors from female patients was that glucose was predominantly oxidized by fatty acid beta oxidation and through the pentose phosphate pathway, which we know generates more moles of ATP than through glycolysis. So, and it also generates a lot of macromolecules for building cancer cells, such as nucleotides. So we had this hypothesis that potentially these tumors were generating, more energy for metabolite generation and cancer cell growth. But there was one pathway which we found particularly interesting that was very different between males and females, and that was in the asparagine synthesis pathway. So within this pathway, aspartate is metabolized in an ATP and glutamine dependent reaction into asparagine, and this is catalyzed by asparagine synthetase. Asparagine synthetase, or ASNS, has been associated with poorer prognosis for glioblastoma patients. And we saw, as you can see, just from these box plots, asparagine and glutamine were much highly upregulated in the tissues from female patients, but not in males. Around the same time that we were doing this research, there was another publication out which showed that asparagine could actually be linked to nutrient depletion, and the increase in asparagine could actually be to increase the uptake of other amino acids such as serine and threonine. And this controls mTOR regulation, protein synthesis, and nucleotide synthesis. So we then looked to see if serine and threonine were also increased in these tumors. And as you can see from these box plots at the bottom, they were also increased just in the female patients and not in the males. And they also seem to correlate quite well with asparagine abundances. So this then led us to the hypothesis that, asparagine is increasing these tumors, to increase tumor growth and survival in females with colorectal cancer, and could potentially indicate a nutrient deplete phenotype. So this was our proposed metabolic model that we came to in around 2020, where we see from our untargeted metabolomics data that the tumors from female patients have this enhanced ATP production driven by fatty acid metabolism. This could facilitate asparagine synthesis and amino uptake in a nutrient deplete condition in the colon. And when I'm talking about nutrient deplete, this sort of indicates as well advanced stage tumors where they may have more hypoxia, potentially less nutrients going to the tumor, and the tumor has to rewire its metabolism to, sustain tumor growth. But we wanted to know what the importance of this really was in terms of prognosis of the patient, because at the moment these are just observational findings. So at the time, Xinyi Shen, who's now a PhD candidate, was an MPH student in my lab. She's super talented. And she wanted to look at the associations between all of these metabolites and prognosis. So she first initially looked at the interaction with sex in the association between asparagine abundances and 5-year overall survival and recurrence-free survival. She did this as well for the other metabolites within this pathway, but we found that only for asparagine, there was this significance. So it seems that sex could be an effect modifier for the association between asparagine and survival. At the time, we didn't really know how to treat metabolite levels in terms of doing these sorts of tests, because there aren't that many studies that have looked at metabolite levels and outcomes in this way. So we chose to do 2 models where we first treated asparagine levels, because they're like concentrations, as a continuous variable. And then we decided to also look at them dichotomized by low levels compared to high levels of asparagine. And what we see here are, sorry for the table, I know it's not very exciting to look at, but we have the 2 different models, model 1 and model 2. We had 5-year overall survival and recurrence-free survival. And what this data shows is that female patients have a much poorer survival if they have high levels of asparagine in their tumors, but there's basically no effect in males. Asparagine, actually in just one of the models, it showed it actually had a better outcome for male patients if you had a high asparagine level. So this was quite striking to us, that this metabolite that we found very high in female patients was actually linked to prognosis. And here are some Kaplan-Meier survival curves just to show the same data really. We have overall survival and recurrence-free survival, female patients on the left, male on the right. And the dark red line is high levels of asparagine. And you can see again that there is poor survival for female patients with high levels of this metabolite. So we wanted to then validate our findings, but we couldn't find another tumor tissue biobank, unfortunately, or publicly available data for colorectal cancer metabolomics. So we decided to look at the transcriptome level, and we know there's a lot of data out there that's publicly available. So we looked at the TCGA cohort, and we looked at the asparagine synthetase gene expression level. So this is the enzyme, so it encodes the enzyme that converts aspartate to asparagine. We first looked at all stages together, stages 1 to 4, for the differences between, well, the association between high ASNS levels compared to low levels and outcomes. And we saw for females, that a high level of ASNS was associated with poor outcome. And then we just looked by stage, and we saw that this was really only for the later stage patients, so the advanced tumors. So a stage 3 and 4 tumor from a female will have poorer survival for the patient if they have high ASNS expression. And notably, again, there was no association in males. So it seems that both the metabolite and the gene that makes this metabolite were both associated with poor outcome. So our next steps with this was to then go into more of the biology of what could be potentially happening here. So we know that ASNS is a ATF4 target gene, and it sits downstream of oncogenic KRAS, basically. So coincidentally, female patients also have a higher incidence of KRAS mutations. So we decided to first go into models where we had a KRAS mutation in our colorectal cancer cell lines. At the time, this was, I think, June 2020, so we couldn't find a collaborator that could do the CRISPR knockdown for us. So we used a company to knock down ASNS in the HCT116 cell line. And then we just basically injected these into immunocompromised mice in a small pilot. to see if there was a difference in tumor cell growth. So we had an N of 5 per group of female mice, where 5 received the wild type cell line and 5 received the knockout cell line. And after 3 weeks, these mice were sacrificed, and we measured the tumor sizes along, this timeline. And we saw that if the mouse had the knockout cell line, they had decreased tumor growth and tumor volume. I'd like to mention too that this work was initially started by Dr. Aladela Kuhn, who's a postdoc fellow in my lab, and he's still working a lot on this project. We looked at some of the histology, and we had a pathologist from the Cancer Center help us with this as well. And we saw that in these knockout cells, there was decreased mitotic activity index, and there was also decreased proliferation as well. So we did the Ki-67 staining here to look at proliferation. So this just indicates to us that these tumors from the knockout, using the knockout cell line in female mice, have decreased tumor growth and are potentially dividing less rapidly. So of course, we then wanted to see if there was differences between the females and males, now that we did see this initial difference, and we just did a survival study. So we had the same cell lines again, but injected now into male and female mice, and we increased the numbers to about 10 per group. And these are just survival curves. So on the top left-hand corner, we're comparing the survival of the male mice that had the wild type on the knockout. You can see there's no differences in survival. If we compare the wild type cells for male and female, there's no differences in survival. But on the right side, we're comparing with the knockout. So the top right-hand corner, we have female mice that receive the wild type on the knockout, and you can see the knockout mice who are female have much prolonged survival. If we compare the knockout from the males to the females again have much longer survival. So even in this mouse model, we can see that ASNS does potentially contribute to cancer cell growth in a sex-specific manner. So, our next question was what could be maybe underlying these sex differences, and I think now these sex differences are really, becoming quite apparent in a lot of disease areas, such as, Alzheimer's and Parkinson's and things. So we wanted to look at the estrogen receptors initially. So we know of the 2 nuclear receptors, estrogen receptor alpha and beta. So estrogen receptor alpha is not expressed in the colon, so we didn't pursue that any further. Estrogen receptor beta is known to be protective of colorectal cancer, and actually its expression is lost in colorectal cancer patients, especially at later stages. And it has been linked to apoptosis and also anti-proliferative states. And then we heard about a novel estrogen receptor, which actually sits on the membrane. It's called GPA, and it's a G protein coupled estrogen receptor. And what's interesting about this receptor is that it's nutrient sensitive. And there's a lot of contradictory evidence in the literature about GPA. And I think this is because they haven't really considered the nutrient status of what they're looking at that time. So it seems that from what we could find in the literature, that it could actually be protective in earlier stages of cancer, and then it could actually have pro-carcinogenic effects in the later stages of cancer. And this could be potentially through its regulation of PI3K and AKT, which sits just upstream of ASNS, and you can see they're all linked to KRAS. So this was kind of our hypothetical a mechanism which was in a review that we published last year. So we were really interested in this nutrient sort of environment within the tumor and how it could be potentially linked to the differences that we are seeing. So in some work led by Dr. Liu, we looked at another cell line, an SW48 cell line, and its isogenic mutant counterpart. And we wanted to see just what the effect was of changing the nutrient status of these growing tumor cells in vitro. So we grew these cells as spheroids, but we decided to have a sort of nutrient deplete phenotype by removing the glutamine And then we had a normal nutrient status by adding the glutamine. So the glutamine replete or the nutrient deplete cells are on the left and the nutrient repleter on the right. And we just compared ASNS expression between the wild types and the mutant cells. And we saw that the mutant cells had much higher ASNS expression if they were grown in this nutrient deplete phenotype, suggesting that they could be trying to recover cell growth, because we know when you remove glutamine, the cells don't grow as well. We then looked at GPA and we saw the same thing. So GPA was also similarly increased in the mutant cell lines compared to the wild type, only in these nutrient deplete conditions. Of interest here as well, we did add estradiol in to see what the effect would be on these cell lines. And we saw, as expected, that they did decrease cellular proliferation, but they had no effect on the mutant cell lines in this nutrient deplete condition. So this signified to us that there could be some resistance in these KRAS mutant cell lines to estradiol's effects when you don't have a normal nutrient supply. So we looked at GPA and TCGA, and we saw the same as ASNS again. We saw that at stage 3 and 4, that if you have a high GPA expression, you have poorer prognosis. But at stage 1 and 2, it seems that GPA is trending towards being actually protective. So we're now kind of seeing this link between GPA and ASNS, which is quite interesting, that seems to be really apparent in females in later stage tumors. And again, there was no effect of, there was no differences seen in the male patients. So just finally, this is kind of the data where we're at right now. This was given to me just last week by my postdoc. We looked at another cell line to see if we could see the same effects. As we know, between cell lines, you may not always get the same finding. So we took these CRISPR edited cells, again, the HCT116 ASNS wild type cell lines. Let's see if I can you see oh yeah, okay. I have a pointer now at the end, sorry. So we have the normal nutrient conditions which we're growing our cells in. So these are the untreated cell lines, and we gave them estradiol, and you can see that this is the spheroid sizes, or the abundance of the spheroids, and you can see that it does decrease cellular proliferation, cell growth rather. But surprisingly, under the nutrient deplete conditions, estradiol this time actually decreased cell growth. So we measured GPA expression in these cell lines, and we saw that GPA was not increased in a nutrient deplete condition in these cell lines. So this actually gave us an opportunity to see what would happen if we tried to increase GPA in these cell lines. So we found this agonist called G1 that can increase GPA expression. So we added G1 to these cell lines, and we found that it actually rescued cancer cell growth. Only under the nutrient deplete conditions. You can see here just how the cells are growing when we add G1 back in, when GPA is expressed. So it seems that GPA can actually work with ASNS and estradiol to increase cell growth in a later stage tumor. And if you see here, these are the same cells, normal nutrient conditions, growing with G1, and at very high G1 concentrations, you actually get less cell growth, which kind of correlates with the clinical findings that this, or the TCGA findings rather, that this could actually be protective in early stage tumors. In the knockout cell lines, we don't see any effect of estradiol on cell growth. So it appears that estradiol is only anti-proliferative in these cell lines when ASNS is expressed. For some reason, we have this high level here of G1 at 0.01 micromolar, which we can't quite figure out right now, but we don't know what the pharmacodynamics are of this G1. So that's something that we're going to look into a little bit more. So just to, summarize really that, our untargeted metabolomic findings kind of, led us to looking at ASNS to see how it's linked to this asparagine production. And we found that, it could be that ASNS and GPA are actually increased together synergistically when the nutrient supply of the tumor is stressed, so at these advanced stages, in a manner to actually recover cell growth and increase cell growth. And it appears that estradiol is really important in this interaction as well. And we see this associated as well with the prognosis of the patient. We have started to look at some of the environmental additive effects, but I'm not going to go into it in this talk. But basically, asparagine is a non-essential amino acid, so it could also come from extracellular sources. And this presentation just shows the intracellular production. So we know asparagine can come from the diet. It can be metabolized and produced by the microbiome and consumed by the microbiome. So we're now also manipulating these sources to see what effect this has on cancer growth. And this is just one slide that shows basically, here are in the green, our knockout cells. If we add asparagine into our media, it increased cell growth. If we add asparaginase into our media, which is an enzyme which converts asparagine back to aspartate, we can see that it decreases cell growth. So we can see that this has effects on the cell line growth. I don't think I have time to go into the bacterial part, but we have. I just wanted to mention we're doing this at the moment in collaboration with a couple of the microbiome groups at Yale, but we have grown 350 bacterial species in vitro, and we've done all of the metabolomics on these species individually. We then took 100 of these and monocolonized them in gnotobiotic mice. We took the SQL contents and analyzed the metabolome of all of these. So right now we're trying to ID novel microbial metabolites that we could potentially find within our colon tumors, because we have all of this data. So we're trying to cross-correlate everything. But within this data, we had a quick look at asparagine and aspartate, and we can see there's many different ratios of aspartate to asparagine in these 100 different bacteria, showing that they do have, different enzymatic activity, potential asparagine producers and consumers. It could also be affecting tumor growth within these cancers potentially. So overall summary, we see asparagine production increase in female CRC patients. This associates with poorer survival. We think that this could be driven potentially by ASNS and GPA that act together to increase tumor growth in advanced stage tumors, leading to a poorer prognosis. So this is kind of where we're at with this project right now. This was really all led from just an agnostic metabolomic study. We didn't know what we were looking for, but then it kind of led us to this hypothesis right now. So I'd just like to thank all the individuals in my lab who do all of this work, and my collaborators at School of Medicine and Sloan Kettering, and then the funders of this work as well.